would like to welcome you all for the first lecture in memory of Daniel Amit, and in particular Daniel's family, Dania, Yali, your son. I'll just say a few words, then we will award fellowships to two students, then introduce our speaker, then we'll have the lecture, and then at the end of the lecture, Dr. Yali Amit will close the event with a few words of his own. The ICC owes much of its agenda to Daniel's pioneering vision, starting in the mid-80s of the last century. It was a time when the boundaries between physics and biology, fortified by cultural and methodological barriers, were not yet transgressed. Nevertheless, Danny realized already then the importance of multidisciplinary research to the study of the brain. What initially seemed like a cursory application of statistical physics to a brain-inspired problem quickly developed into a lifelong commitment to the discovery of the theoretical principles underlying the collective aspect of brain functions. Danny's work and articulated vision helped propel neural computation into mainstream neuroscience and transformed multidisciplinarity in research and education from an anomalous esoteric into the gold standard of brain sciences. However, Danny was not just an excellent scientist. He was a thinker, a forceful human rights advocate, and a political activist, an ardent reader, and eloquent writer. We did not always agree about the interference between these domains of activities, but he was and will always remain to his friends, colleagues, and students a role model of a true intellectual, a scientist with the unique ability and courage to explore the broadest horizons of humanity. Let me quote from an article that Danny wrote to Liberismo, an article which I received in its Hebrew translation by you, by Dalia, I think, and I retranslated it into English. Just one paragraph from Danny's article. Science must encourage the dialogue with other forms of knowledge, and above all, with the conscious, ecological sensitivity, and religious ethics, not to persuade people to go to church, although this is not such a bad idea given the conventional forms of pastime activity, but to enrich life with other experiences that will guide us in choosing which research to carry out, and to scrutinize the social models underlying the funding of science. These issues need to be examined by criteria which are external to science itself. So this is one symptom of Danny's broad vision, not only of science, but also of ethics, morality, and the plight of a human being. So, as I said, we will tonight honor Daniel Amit's memory by awarding two fellowships to our outstanding students. The first one, Dimitri Davidov, will receive the achievements in the study of computational neuroscience, and more specifically, to his excellent research in language processing, particularly the concept formation of language, supervised by Dr. Ari Rappaport in computation in the computer science department. The second award to Shaul Bruckmann. Achievement in the study of models of interneurons in a cortical column, more or less supervised by Professor Idan Segev from the Department of Neurobiology. To introduce our speaker, Professor Yasushi Miyashita is a world leader in the field of functional architecture and neural mechanisms of memory in the cerebral cortex. Professor Miyashita received his PhD from the Department of Physiology in the medical school, the School of Medicine at the University of Tokyo in 1978, and he became a full professor of the same department in 1989. He's a recipient of many awards and honors, including recently the medal with purple ribbon from the Cabinet Office Government in 2004 and the Japan Academy Prize in 2007. Miyashita's work had a long-lasting influence on Dani. His 1988 Nature letter on neuronal correlates of visual associative long-term memory in the primate temporal cortex immediately caught Dani's attention, because Dani saw this as an opportunity to form a testing ground for the attractor model of associative memory. I remember Dani's visit to Miyashita's lab in Japan. He was very excited about it, and he came back and he told me, I don't understand those neuroscientists. They publish a Nature letter with 3 figures, and then they have all this enormous amount of data in their drawers without publishing it. He was very fascinated by what he learned from Professor Miyashita. This led to a long-term experimental and theoretical research agenda together with Charles Rockstein, Udi Zohari, Valodia, and their colleagues, and served actually as one of the first successful attempts to bridge the gap between network theory and experiment in the field of computational neuroscience. We are therefore very happy that Professor Miyashita agreed to come here and deliver the first lecture in memory of Daniel Amit.
Okay, thank you for your kind introduction. Yes, it's my great honor to be here and to deliver this Daniel Amit Memorial Lecture. Daniel Amit was a great scientist and my friend. I first received a letter from Daniel Amit in 1989, actually. Okay, not 1988. 1989, when I published 2 Nature papers on visual short-term memory and long-term memory. I remember I was a bit surprised to receive such a letter from famous computational scientist, because at that time I myself did not understand theoretical or computational implications of my discovery. In 1991, when I published a third Nature paper on temporal association mechanism of long-term memory, Dani gave me a long letter with detailed analysis of attractor properties of his neural network, which captured really some very essential part of my experimental findings. Honestly speaking, I understood the importance of his computational framework first at that time. Since then, we exchanged a number of letters and ideas. Yes, Daniel Amit was a person with extraordinary passion. He gave me a lot of ideas on interesting but very difficult novel experiments, which I could actually run only 2 or 3 of them in 6 or 7 years, perhaps. And even after my own scientific interest went somewhat away from local circuit of the temporal cortex, Danny gave me lots of novel inspiration. So his passing away is a great loss. Today, I brought mostly topics on local neuronal network in memory of Daniel Amit. And that's the last part of my lecture. I want to talk some open problems we are now struggling in the experimental analysis of local circuit. I wish to hear what Danny would have suggested me if he is here. Okay. So, oops, yes. So here, oops, something wrong. Color is wrong. Won't you help me? Okay, that's good. Okay. Yes, so that's a cover letter. Then something wrong? Right. Yes, okay, good. Thanks. Then first, let me visualize the target of my talk. Okay, oops, no, okay. Oops, back. Oh, okay. Is it better? Which is better? This is better, or which is better? Okay. Okay. So let me first visualize the target of my talk. That is internal representation of visual objects in the temporal cortex. Actually, Chaim told me today. First of all, I must say, where is the temporal cortex? Okay, so I'm saying, here is the temporal cortex. Okay, and although I'm also interested in top-down neural process that activate this internal representation, okay, I will not talk about the top-down signal mechanism today. The core message of my talk is that internal representation, okay, is associatively organized, and the associative link is mediated by a group of memory neurons called pair coding neurons. I will discuss later how these schematic diagram can be understood more realistically at the level of neural circuit. So here is the menu of my talk today. I start from the topics of mnemonic representation of objects in the temporal cortex, particularly in area T, area 36, and area 35 of the temporal cortex. Then I will discuss empirical or experimental evidence on local circuit and cell assembly, okay, and some recent and published data on complex information flow between temporal cortical neurons. Okay, first let me start from a very, very basic problem. What's the issue? I have been demonstrating that in monkeys, memory representations of objects are created in the temporal cortex after initial memory encoding into hippocampus. However, in humans, it has not been exactly identified whether or where long-term representations can be created in temporal cortex after initial consolidation process in the hippocampus. So here, I want to start my talk from evidence on whether and where the long-term representation can be created in humans in forward experimental study. Okay, as I noted, human long-term memory has been characterized as associative storage for semantic and episodic events. So I have been investigating basic memory mechanism of associative storage by using a cognitive memory task called pair association task. Many neuropsychological clinical batteries contain this task as a useful tool, for example, in detecting mild memory impairment in early Alzheimer's disease. So please have a flavor of the task first. These stimuli are called visual paired associate, and as you see, these 24 pictures are arbitrarily sorted into 12 pairs. And humans and monkeys must memorize these pictures as well as pairwise associative relations of these pictures. For example, this is the pair number 9, and it consists of two pictures. One picture 9, number 9, and the other picture number 9 prime. So when the subject is given one picture of a pair, for example, this picture, he must retrieve from his memory the other member of the pair, that is this picture. Okay, clear? Then, please have a look. Then, for example, this is a Q picture. Okay, this is a Q. Then, which is a paired associate of the previous Q stimulus? If you remember the pair, you can answer. Okay, this one is the paired associate of the previous Q stimulus. Can you see? Okay. So let's return to the experimental protocol of the human fMRI study. Human subjects initially learned set of picture pairs, these picture pairs, through a trial and error practice of pair association learning, just as a monkey did. So in the task, each trial started from the presentation of a picture as a Q, and then presentation of the two pictures, one as a target and the other as a distractor. And then feedback was given whether it's correct or not. The subject learned picture pairs in this way until performance criteria. Here is the timetable of the task. Subjects initially learned 10 pictures. This is the remote set. Then approximately 8 weeks after the initial learning, subject learned another set of 10 pictures. Okay, these are recent set. And the performance of these two picture sets were balanced by more intensive repeated training to the remote set. It's quite important to make the performance of these two sets balanced, because the resultant fMRI signal may be masked by a retrieval success effect if our performance is different. Okay, following on the practice, functional MRI, okay, was used to compare brain activity during retrieval of the two picture set, remote set and recent set. Okay. Then, prominent activation was observed in multiple regions in temporal cortex, in the contrast of remote set minus recent set. Activation included anterior temporal cortex as well as perianal cortex in both hemispheres. And this anterior temporal activation, okay, was a significant correlation with the reaction time in choice period, indicating a behavioral impact of this activation. It should be also noted that in the opposite contrast, that is recent set minus remote set, activation was observed in the hippocampus as expected. So the results provide direct evidence for development of cortical representation in humans. Okay, that's okay. But spatial resolution of fMRI is not satisfactory in examining microstructure of this presentation. So in monkeys, more details are known about the functional architecture on the basis of single unit recording. Although today I have no time to show evidence we know that perirhinal or anterior temporal representations are localized in multiple patchy cell clusters, the diameter of which is about 1 millimeter, and the distance of which are sometimes less than 3 millimeters. So we conjecture that the temporal cortical representation in humans also consists of similar multiple patchy localized modules. And now I will describe what kind of memory neurons are contained within these localized patches. Okay. So let me return to the monkey study and a little bit back to the past in memory of Daniel Amit. The starting point of my research and the starting point of my friendship with Danny was the discovery of memory neurons that encode the associative relation of the paired pictures. This slide illustrates two examples of such memory neuron that we call pair coding neuron. Upper panel indicate rastergrams, and lower panel indicate peristimulus histograms. And these two neurons were strongly activated during the presentation of one stimulus called optimal stimulus. Okay. And the paired associate of the optimal picture evoked the second strongest response. So here are quantitative stimulus selectivity of the neurons shown in the previous slide. Ordinate shows the response to each stimulus, and abscissa indicate the pair number, and open and closed bars corresponded to the paired associate of each pair number. So this neuron A has a very sharp stimulus selectivity and fired only to picture number 4 and picture number 4 prime, and did not respond to other stimuli at all. This neuron B has a broader stimulus selectivity, but fired mostly to picture number 5 and picture number 5 prime. Therefore, these neurons represent which two pictures are combined into pairs, and therefore encodes the semantic linkage between the paired pictures. We then systematically surveyed these neurons in inferotemporal cortex. It is known that the inferotemporal cortex consists of two major subdivisions. One is the neocortical subdivision, including area T here. That's a lateral view of monkey brain, frontal cortex here, occipital cortex here, parietal cortex here, and the temporal cortex here. And area T is located here. And the other division of the inferotemporal cortex is a limbic subdivision, including area 36 of the perirhinal cortex. Here is a ventral view of monkey brain, and the coronal section cut at this level is shown here. So you see, ARTE occupies more lateral part of the inferotemporal cortex than area 36 does. We systematically investigated pair coding neurons in the inferotemporal cortex and found that both area 36 and TE represent pair association memory, but more prominently in area 36. Here is the population averaged responses of neuron in TE and 36. When normalized with the optimal response to each neurons, the response to the paired associate of the optimal stimulus was quite large in area 36, but response to the paired associate was a bit smaller in area TE, but still significantly larger than other responses. And this tendency was further quantified by using an index that we call pair coding index, PCI. Pair coding index is actually a correlation coefficient between the responses to these picture pairs. Here is a population distribution of the pair coding index in area 36 and TE. You see the distribution was significantly shifted positively in both area 36 and TE, and the median value was 0.14 in area TE and 0.51 in area 36. So neurons in area 36 and TE were very different regarding the pair coding memory. Physiologically important point is whether this difference in pair coding index arises from difference of other general stimulus selective properties. So here we calculated two different selectivity indexes. One is the R square, which provide an estimate of how much of the variance in firing rate can be accounted for by the factor of stimulus. Another is a tuning index, actually a kurtosis, which is a measure of the sharpness of stimulus selectivity for each neuron. Okay. As you see, in these cumulative distribution histograms, the distribution of either R square or kurtosis did not differ between two areas. The left panel demonstrates, for your reference, the distribution of the pair coding index, which is clearly different from area 35 and TE. So here is the second interim summary. So pair coding neurons provide linkage between operationally related but physically unrelated objects. So it's a pair coding memory, neurons. Second point, associative property of single neurons proceed in the inferior temporal cortex from area TE to area 36. Then let me proceed one step further along a bottom-up pathway. Area 35, okay, that's a medial subdivision of the perianal cortex, is cytoarchitectonically distinct from the lateral subdivision, area 36. Functionally, however, area 35 has never been studied in behaving monkeys. One plausible reason is the location of area 35. Please have a look. Area 35, okay, it's a coronal section at this level, okay. Area 35 is indicated as a red area here, because area 35 is a narrow strip on the fundus of the rhinal sulcus here. It has been very, very difficult to systematically record single unit activities from this area in behaving monkeys. We challenged a recording from area 35 using a new MRI-based approach. Before the experiment, we made MRI detectable metal deposit markers, okay, just above area 35 using an L0 microelectrode. The location of the metal deposit, okay, were determined using both X-ray and both high-resolution MRI. And in daily recording session, the position of the microelectrode were determined on X-rays, overlaid on MR images, using this metal deposit as a common reference. We further confirmed the recording site using several electrolytic lesions, which are made within area 35 or its border, and then histologically visualized. Okay. With this method, we recorded from 181 neurons in area 35 or 2 monkeys while they performed the pair association task. 61 neurons showed stimulus selective activity, and they tended to cluster within area 35 along the anterior posterior axis at this level, as shown by closed circles. Okay. Here are similar data from another monkey. Also, here is the electrolytic lesion, and the selective neurons are clustered here at this level again, within a very small area, patchy area. Then we analyzed pair coding properties of area 35 neurons, just as those of area 36 neurons. Pair coding index, PCI, which I defined just before in my talk, did not differ between area 35 and 36. In both areas, neurons with selective delay activity showed more prominent pair coding property, than those without selective delay activity. This was a common property. So pair coding property itself was a common property between area 35 and 36. But we found that area 35 delay selective neurons had a very distinct property. These are two representative delay selective neurons in area 35. One stimulus elicited a strongest response. Okay, that's optimal stimulus. Okay, these are two different neurons. That's optimal response. Then in these neurons, the paired associate, okay, of the optimal stimulus evoked responses just comparable in amplitude with those in the optimal trials. In contrast to the robust responses to the optimal and its paired associate, okay, these neurons responded only negligibly when a stimulus from other pairs were presented. We quantified these tendencies by an ROC analysis, receiver operating characteristic analysis. With ROC analysis, we estimated each neuron's performance to discriminate the presented stimuli. As shown in the left panel, okay, the areas under the ROC curve called AUC values were significantly smaller in area 35 neurons than those in area 36 neurons when ROC curves were drawn so to discriminate the optimal stimulus from its paired associate. If we call the pair of optimal stimulus and its paired associate as the primary pair, okay, the result of the left panel, okay, can be said that AUC values for discrimination within primary pair is significantly smaller in area 35 than area 36. On the other hand, AUC values that discriminate the primary pair from other pairs were not significantly different between area 35 and area 36. Okay. So these results demonstrate that a group of neurons within area 35 had a significant tendency to represent paired stimuli as a single item. We call this as unitization processing. We therefore propose a model in which the unitization occurs during the step from area 36 to area 35 along the hierarchical organization. In area 36, okay, object association process proceed, but in area 36, both objects in the primary pair are still discriminated. In area 35, neurons do not discriminate unitized object, but of course, discriminations between pairs are still preserved. Okay, that's hierarchical processing within the temporal cortex. Okay, now let me move on to the analysis of local circuit and cell assembly in area 36. Okay, I'm now returning back to area 36. First step is to construct a simple model of local circuit that can create pair coding memory neurons. I started from scrutinizing the empirical data. In this population average of responses of area 36 neurons, the paired associate, okay, of the optimal picture evoked the second strongest response, okay, which is a hallmark of pair coding memory response. When we look at the response time course in more detail, okay, the amplitude of initial transient response to the optimal stimulus were much larger, okay, than those of paired associate, but the amplitude of the late response, okay, were relatively similar. So we hypothesized that this may originate from two different groups of neurons and analyze the time course of the response in each neuron in terms of two indices, half peak time and early response time. Then the two indices of each neuron are plotted on this scatter diagram, and cluster analysis reveals that these area 35 pair coding neurons are separated into two different classes, type 1 pair coding neuron and type 2 pair coding neuron. In type 1 neuron, okay, a time course of pair response and optimal response are quite similar, but in type 2 neurons, the responses to the paired associate, okay, lacked initial transient and exhibited only sustained activity that developed after a delay of tenths of milliseconds. Then, inspired by a conceptual framework by that Donald Hebb presented in his book Organization of Behavior. Okay, these figures are taken from the textbook. I have drawn an essentially identical sketch in the right panel that clarifies that pair coding neurons should be shared as a common element in the two reverberating networks, each of which represents picture A or picture B. Then I would like to suggest specific positions of type 1 neuron and type 2 neurons in this diagram. For example, type 1 neuron, okay, here, okay, encode both pictures by direct convergence, okay, from two single coding neurons. In contrast, type 2 neurons, okay, may be driven directly from one picture, but only indirectly from another new picture. Then the question is whether this model can be experimentally testable or not. I believe it's testable by a spike correlation analysis by simultaneous recording from these neurons. Okay, before going into model testing itself, we have run some methodological investigations about dynamic connectivity of simultaneously recorded multiple single neurons in the monkey temporal cortex. In this experiment, we recorded multiple pairs of neurons simultaneously using heptode or tetrode, or heptode. Here is an example of spike activity recorded simultaneously in each of 4 channels of the tetrode. Okay, you see, just like that. When multiple pairs of single neurons were recorded in that way, we found that their connectivity changed dynamically depending on various contexts, such as stimulus or task events. Here are examples we initially encountered using an easier behavioral task, visual discrimination task between face-like objects and non-face-like objects. Briefly, here are cross-choreograms and peristimulus time histograms of a representative pair of neurons that's recorded. These are cross-choreograms of this pair to the optimal face-like object or non-face-like object. Abscissa indicates time lag of spikes of cell 1 and cell 2, and ordinate indicates spike count. Gray traces here indicate shift predictor that were calculated from shuffled spike trains. So lower traces, okay, indicate shift predictor subtracted choreogram. So this neuron pair exhibited a significantly correlated spike firing only to optimum face-like objects, not in non-face-like object. Okay, so we must cope with this kind of dynamic stimulus dependence in the experimental test of the model of pair coding neurons. First, we tested a model prediction that type 1 pair coding neurons receive input from single coding neurons with corresponding stimulus selectivity. Because the connectivity would depend on the context, we must test the connectivity for each stimulus separately. Here are examples of spike density histogram of simultaneously recorded area 36 neuron pairs. Okay, so these are the pair stimulus time histogram for unit 1, and these are the stimulus time histogram for unit 2. You see, this is the PST histogram for stimulus number 2 in unit 1, and this is the peristimulus time histogram for stimulus number 2 in unit 2. So both unit 1 and unit 2 are highly stimulus selective neurons, you see. And unit 1 is single coding for stimulus number 2, as you see. And while unit 2 is type 1 pair coding neuron for stimulus number 2 and number 2 prime. Okay. Then here are raw cross choreograms and shift predictor subtracted cross choreogram between unit 1 and unit 2. The shift predictor subtracted cross choreogram, okay, exhibited a displaced peak indicating connection from unit 1 to unit 2, which is consistent with the model prediction. Here is another example of simultaneously recorded neurons, but here we could isolate 5 different neurons in the same recording session. Here are examples of spike density histogram of 2 of the 5 neurons. And unit 3, okay, unit 3 is single coding for stimulus number 5. And unit 1 is type 1 pair coding for stimulus number 5 and 5 prime. So here are raw choreogram and shift predictor subtracted cross choreogram between unit 1 and unit 3. So you see the shift predictor subtracted cross choreogram exhibited a displaced peak indicating a connection from unit 3 to unit 1. So stimulus selectivity and the connectivity among the 5 simultaneously recorded neurons are summarized here and schematically drawn. In the cross-correlation analysis of the previous slide, we confirmed the connectivity from unit 3 to unit 1 in the context of stimulus number 5. Similarly, we also confirmed connectivity from unit 4 to unit 1 in the context of stimulus number 1 prime. Therefore, so far as we have tested, connectivity in each stimulus configuration is mostly consistent with the prediction or with the model. However, at this stage, it's clear that in this model, there is no gating mechanisms that control the connectivity depending on the stimulus context. For example, how the dynamic connectivity from unit 3 to unit 1 is made effective only in the context of stimulus number 5. I'm sorry, I don't know the answer yet, but we recently obtained another lines of evidence using, again, visual discrimination tasks, suggesting that actual local circuit dynamics can be far more complex in monkey temporal cortex than we assumed in the simple model. in this representative pair of neurons, cross-choreogram, okay, exhibited a displaced peak, okay, indicating connectivity from unit 1 to unit 2. Okay, that's a suggestion from a cross-choreogram. Then we run a spectral analysis. So here is a coherence analysis. The coherence analysis also demonstrated gamma range communication between unit 1 and unit 2. And statistical significance, okay, set here, okay, of the coherence was evaluated using a new distribution of the data calculated from trial shuffled spike trains according to the method by Pascal Fries. Then we ran non-parametric version of Granger causality analysis and found that Granger causality, okay, revealed prominent peaks at a frequency range similar to that of coherence. It is also clear that the peak causality values in the direction from unit 1 to unit 2, okay, so red is causality from unit 1 to unit 2, okay, was much greater than that in the opposite direction. That blue ones is a causality from unit 2 to unit 1. And this dominance of direction from unit 1 to unit 2 was consistent with the direction indicated by cross-choreogram. Statistical significance of Granger causality was evaluated using trial shuffled spike trains, as in the case of coherence. The directionality of the Granger causality was defined as a difference in the causality value between the two directions. Okay, so this is a difference between the two. Okay, here are population data from 21 cell pairs that exhibited a displaced peak on the cross-choreogram. Upper panel shows the spectral properties of spike trains during the Q period. Okay. And here, if the cross-choreogram indicated connection from unit A to unit B, then direction from unit A to B was defined as forward direction. That's a definition of forward direction. Then both coherence and Granger causality was greater than the trial shuffled control in the Q period. Particularly at the gamma range, you see. Directionality of the causality, okay, also revealed a prominent peak in the gamma range. Lower panel shows the same spectral properties during the baseline period before Q stimulus presentation. In the baseline period, both coherence and Granger causality was much weaker than that in the Q period. The statistical significance of these observations on gamma coherence and gamma causality were confirmed by ANOVA, as shown here, although I do not go into details. We then examined the temporal dynamics, along the time domain, temporal dynamics of causality using the sliding window technique in a 125 millisecond window with 25 millisecond step. Okay. Here are results from a representative cell pair. This cell pair exhibited gamma coherence, throughout the Q period. Okay, Q stimulus was presented during this period, so gamma coherence continued during the Q stimulus presentation. Okay. On contrary to this, Granger causality of this cell pair revealed an early transient increase with the dominance of the forward direction. In the late epoch, a transient forward causality reappeared again. Directionality analysis, okay, here, confirmed that forward dominant transient causality appeared twice in the Q period. Firing rate dynamics, okay, firing rate here, of this cell pair did not reveal the corresponding double peak structure at all. Then, here are corresponding population data for the 21 pairs. You see essentially the same temporal dynamics. Population gamma coherence revealed a sustained increase throughout the Q period. Compared with the coherence, okay, forward causality exhibited more transient dynamics, okay, compared with the coherence, okay. This early transient component appeared relatively in high frequency range, and it quickly attenuated, then again reappeared in later epoch. Okay. In contrast to the forward causality, okay, the backward causality exhibited a small and slow increase when the early component of the forward causality was attenuated, and it occurred only relatively low frequency range. Then we quantify the dynamics of gamma component for the coherence and causality. It's clear that gamma coherence, okay, revealed as a sustained increase throughout the Q period, while causality exhibited more rapid dynamics changes in both forward and backward. So red is forward causality and blue is backward causality. So this result may have several implications. A classical view teaches us that a significant displaced peak on the cross-correlogram suggests a posisynaptic serial connections. However, in our recording, such neurons, okay, with displaced peak in the cross-correlogram, such neuron indeed showed granular causality in the gamma range, of whose directionality was consistent with the cross-correlogram. However, temporal dynamics of the causality exhibited complex triphasic pattern. That is, a transient forward component was first appeared, and then it was followed by slowly developing backward component and subsequent reappearance of the forward component. And this complex pattern cannot be easily explained from simple serial connection. Okay, now let me rush on to conclude my presentation. So connectivity between simultaneously recorded single coding and pair coding neurons was mostly consistent with the model on type 1 pair coding neurons. That's good. Because we do not have enough data on connectivity between type 1 and type 2 pair coding neurons, we cannot evaluate that aspect of model prediction yet. Finally, local circuit dynamics may have very complex triphasic go and back type information flow. So a very simple model, which I have drawn, may be unable to capture such aspect of network dynamics. I'm quite eager to hear your views, views of scientists in the field of computational neuroscience, how to understand this situation. I hope we will exchange our inspiration, just as late Daniel Amit once gave me inspiration and enthusiasm in my early research on associative memory. Thank you for your attention. Okay, here is my, here's the list of my collaborator.
And for a few questions.
Regarding the representation to expect, if you have a pair association, of course, what you have to do is retrieve from one image its pair to associate. So really, this task would lead you to a generation of association between images. While when you use the same images, and a task is a discrimination task, what you have to do now is discriminate between the two images. So you have to generate different representations for these two images. Now, if I got you correctly, I think that what you were pointing was that at one area, I don't know if it was 36 or 35, you have a generation of a representation for both, while in another, you have for the difference between the two. Now, does that change depending on what is the task, or is this something that has to do with the anatomy?
Okay, yes, good. But before answering your question, I must say that in area 35, okay, the neurons do not discriminate two stimulus, optimal and its paired associate. But the neurons do discriminate the pairs, okay, the primary pair and the other pairs. So still discriminability preserved. So the specificity of the area 35 neuron is that they may be able to treat the pair, okay, as a unitized item. And so the downstream from area 35, area 36, there are some associative process is going, but still there are some discrimination between. And if you go further downstream in area TE, there are lots of differential representations between these. So I suppose the whole brain can use these representations, say representations in area TE, as well as representations in area 36, as well as representation in area 35, depending on the purpose of the task, I think. So the area 35 can be very, very specialized to solving the task, because it's really the close neighborhood of the hippocampus. Yes?
I guess it's a follow-up on this question. Imagine that you ask the monkeys, after teaching them the pair, to discriminate between the two pairs, the two members of the pair. And then you look at that neuron.
Okay, confused. But of course, I can imagine. And yes, so the neuron will be differentiated again. But I think it will take time. As far as I know, the change of representation in the hippocampus was quick, rapid. But the change in the representation in our perinal cortex takes some time. So although this is a speculative answer, perhaps the neurons will become discriminate the stimulus, say after, say 8 weeks, I don't know. I guess. Yes?
But we can switch from one task to the other immediately.
Yes,
right?
okay.
So going back to Aisha's question, how do we do this?
Okay, so that depends on task framework, okay. I never tested, but perhaps neurons in area TE can cope with that kind of situation. I don't know.
I have a question,
yes.
a global question on these results. What do we know about the causal impact of those representations on the actual processing? Okay.
Yes, initial evidence is that lesion experiment in monkeys actually. That was done by Bettemari and Mott-Mishkin in 1993, I think, with lesion of that part, okay, actually, that lesion include both area 36 and 35 and entorhinal cortex, I think, that has actually prevented monkeys to learn the paired associate. So that has causal impact.
But this is a gross lesion.
Yes, gross lesion, okay.
hippocampus also?
No. Betsimari and Mishkin's lesion included only periorbital, not hippocampus. So it's a selective region.
Your own belief or speculation, or whatever, that these representations are essential for performing a task, because I think at the initial results that you had about delay to match task, the actual performance in delay to match task did not rely on having a long-term memory at all. The monkey performed as well for newly presented pairs.
Okay. Yes. As I told you, the learning of the pair associate, okay, heavily rely on periorbital cortex. I think that's true. But perhaps the matching to sample task can be solved in a different strategy if the stimulus, I don't know. Actually, Betsimari ran a very clever experiment with lesion. And actually, the performance of monkey depends on the stimulus set size. Okay, if the stimulus set is very large, just like say trial unique, the lesion produces a deficit. But if the set size is small, the lesion effect is quite mild. So that will suggest behavioral impact depend on the monkey strategy, how to use the stimulus, trial unique, or say some very well-known sets. But that's a speculation from lesion experiment.
So I think
in Chicago in the winter of 1992, and then I noticed that everything he wrote, which he tried to encourage me to read, despite my resistance, always mentioned these several papers that you referred to. So it's really great that you were able to come and talk in this memorial lecture. I'd also like to thank the people who organized this lecture, Chaim, whoever else was involved in the organization of this lecture, and also the close friends in the Institute for giving these young students this fellowship and enabling them to continuing this interesting study in such a wide and diverse field that I guess spans from programming languages to mathematical modeling of networks to experiments that now are using the most sophisticated statistical tools available. So really thank you everybody, and for me it was a very special lecture and event. Thank you.